John Gribben, who you already met because of his dancing skills at the back of the room, also makes a living as a professor of experimental cancer medicine and director of experimental cancer medicine and director of stem cell transplant at St. Bartholomew Hospital at Queen Mary's School of Medicine, the University of London. So after completing his medical degree, he was a research fellow in hematology at University College London. He was a leukemia research fellow, recipient of the Wellcome Trust Fellowship Award. He's won international awards in postdoctoral work, worked at Dana-Farber also at Harvard Medical School. He's on the faculty at Harvard Medical School and also at Dana-Farber and Brigham and Women's Hospital, and all of that until he moved to London in 2004. And he's a founding member of the CLL Research Consortium, and I think the only one from across the country. Thanks.
I also want to add my thanks to Tom for what he's done. You may be sitting here thinking that it's completely normal for people in different universities and across different continents to be working together collaboratively, but this is a pretty unique. And under normal circumstances, we'd be competing with each other to publish these papers. And it's a testament to Tom's skill that he's held us together for this long, and that we've become very fast and very good friends and very good collaborators. And I think, as he already said, the consortium means so much more because we're able to do so much more by working together than we would individually. So with that said, a little couple of disclosures that you're supposed to, I think I'm the only one who remembered to do that today, but you're supposed to do this every time. Also thought, of course, that for all the rest of us, we thought that Andrew Reeves' job was to tell Tom where he's supposed to be at any time of the day, but I guess he left that part out of his So you've had your master's degree in counselling, you've got your medical degree in CLL, you've had a little bit of coaching in molecular biology, and in 20 minutes you're going to get your PhD in immunology, which is what we're talking about today. So as Tom already alluded to, and other people have alluded to today, CLL is a disease in which we know there's a very dysregulated immune system. Almost for as long as we've known about this disease, we've known that patients have an increased incidence of infections. And the weird thing about it is that in addition to that increased susceptibility to infections, there's also these phenomena of autoimmunity. So what that's telling us is that we've got a disordered immune system, and there's something wrong with the accelerator, and there's something wrong with the brakes. And that's all part and parcel of what an immune system is supposed to have. It's supposed to have accelerators, it's supposed to have brakes, and I'll come back to talk about that issue later. We've also been hearing about, from some of you receiving your IVIG infusions, but of course another hallmark of this disease is that hypogammaglobulinemia. The hypogammaglobulinemia is due to low levels of immunoglobulin in the blood. It's mostly the IgG subtype. The severity tends to increase with time and stage of the disease, but most patients with CLL at some point in their disease will have low levels of immunoglobulins. And even, as we've also already heard, even when it's successfully treated, you can be left with very low levels of immunoglobulins that can lead to repeated problems with infections later on. The precise causes aren't very clear, but it's likely due to interactions with the malignant CLL cells with the normal cells of the immune system. There's loss of plasma cells, there's loss of T cell help to be able to help the normal B cells make antibodies. And we know that there's a direct relationship between low levels of immunoglobulins and very specific infections. And of course, Neil already alluded to the fact of things like making sure that your vaccinations are up to date with things that you want to protect yourself against. Now, despite the fact that we all know that patients have got hypogammaglobulinemia, and despite the fact that many of us who treat our patients with IVIG, insurance companies don't seem to quite agree that it's quite as important as we all think it is, and we often have difficulty in obtaining this for our patients. So we know that CLL affects the whole immune system. We know that it's the CLL cell itself that drives that. And what I'm going to try to take you through is how we see that it has effects on your T cells, has effects on your natural killer or NK cells, as if that there are strange things that the CLL cell does in interaction with these T cells and NK cells, and of course with microenvironmental effects. And my lab and Tom's lab and many other researchers in the CLL consortium spent a lot of time trying to unravel these complex interactions that occur between different cells within these microenvironments. Now, there's something wrong with the immune system. That something wrong with the immune system is because of the CLL cell, and we think a large part of what's happening is that the CLL cell is, to all intents and purposes, switching off your own immune system so that your own immune system doesn't attack the cancer. Now, it's a very attractive proposition for anyone who's got cancer to believe that your own immune system could actually be what's used to treat and cure the cancer and get rid of those last cells. But the CLL cell, we know, has evolved very effective ways to prevent immune attack. And if we want to be able to turn that immune system back on and have your immune system be able to attack the cells, we have to very fully understand what was wrong with the cells in the first place. How does the CLL switch those cells off, and how can we switch them back on again to attack your cancer? Now, this is something called gene expression profiling, and this was work we did. Well, this is actually work published while we were still at Harvard from samples from the CLL consortium. And each one of these columns is an individual patient, and each row is an individual gene. And what we're looking at here are differences between age-matched healthy individuals and patients with CLL. And here we're not looking at the tumor cells, we're looking at either the CD4 or the CD8 T cells or the NK cells. Now, most people who are looking at cancers look at the genes that are different between the cancer cell and a normal cell. Here we're looking at cells that are supposed to be normal compared to those of an age-matched individual. And without your doctorate of immunology yet, you can clearly see that these do not look normal. And if you want to choose the most expensive way to diagnose CLL, you could do it by doing gene expression profiling of your T cells, but we wouldn't probably want to do it that way. But what that did was give us lots of clues as to what were the ways and the pathways in which these cells were abnormal, and try to get to the bottom of what had caused these defects in the first place. So put very simply, here's what's supposed to happen. That's a T cell that's on the right-hand side of the slide here, and that's what's called an antigen-presenting cell. That's the target cell of the immune cell. And what's supposed to happen is that T cell is supposed to latch on and form a very tight connection between the T cell and the cell that it's trying to kill. And here's what happens in CLL. There's lots of that protein on the surface of the T cell, but you'll see it can't bring together, and it can't very effectively kill that target CLL cell here. So there are multiple gene defects that we know are induced by the CLL cells, and I'll come back to talk about that in a moment. Because these proteins don't come together at the surface, there's impaired ability to see these foreign proteins, some of which, of course, Tom's already shown you in his wonderful little video there, proteins like ROR1, things that are uniquely expressed on the CLL cells that your immune system should be able to see. And what that does is it results in very poor functioning T cells. So poor T cells in terms of not having good responses against infection, but just as importantly, poor T cells in terms of an inability to see and attack your CLL cells. Now, the other thing we see is that the T cells in the patient's blood, and again, these samples that Laura provided for us through Project 115, probably, of the CLL consortium, is that we see that the types of T cells which are in the blood of patients with CLL are again very different from what we see in age-matched individuals. What we should have is we should have a lot of our cells that are what are called naive. These are cells that have an increased ability to see new infections or new things which appear in your blood. And what you'll see is that in patients with CLL, these cells are very depleted. And instead what you've got is a big expansion of what are called antigen-experienced cells, kind of, put it another way, kind of clapped out cells that have kind of almost given up the ghost and aren't able to do very much. And that's exactly what we call them. We call them exhausted T cells. What's happening there is that these T cells are chronically being stimulated, likely by the CLL cells themselves, to come into this just exhausted state. And what we're looking for is the ability to repair these T cells and turn them into cells which are more able to affect what we're looking for in terms of having an immune response against the cancer. So how are we able to do that? Well, you can take your clapped out old T cells and you can replace them with somebody else's. That's what we're doing with an allogeneic transplant or using donor lymphocytes. There are immunomodulatory drugs like lenalidomide and now CC122 that are able to repair a lot of the defects, and we've done a lot of work demonstrating what those mechanisms are. I'm going to give you a little introduction into what checkpoint signals are towards the end of this talk. So there's the PD-1, PD-L1. We were first discovering this to be important in CLL, and Bill Weirda and I and others have been trying and knocking on the doors of companies for years to try to convince them that CLL is one of the diseases in which you should be studying this instead of the bigger solid tumour market that they've been looking at instead. There are a whole variety of different novel antibodies, and of course right at the bottom here, of course what you can do is soup up these T cells using the CAR T cell approach, and I'll talk about that a little bit also. So let's just talk very briefly about CLL and transplantation. So allogeneic transplantation has been viewed as being a reasonable treatment option for very poor risk CLL. And the transplant community has quite well-defined criteria of what we mean by disease that has a bad enough prognosis to consider a transplant. That's patients who have non-response or early relapse, relapsing early after combinations, or patients who have p53 deletions or mutations, and I'll take you through all of this. So what's the data that tells us that these are the groups of patients that we should be having? Well, here's the data looking at the overall survival of patients that is generally short in those who don't respond to first-line chemotherapy. This is data from Bill Weirdas from MD Anderson. What you're seeing here is, of course, that although this is where you want your response to be if you're going to have RFC-type therapy, there are individuals who instead do this, and these people are going to do poorly, or at least we're going to be doing poorly. About 30% of patients will have a rather shorter survival after this type of therapy than we're looking for, and it's how do we identify who these patients are so that potentially we don't offer them that therapy in the first place, or try to find other ways to salvage them back. The other group of patients that we know don't do well are something that Dawn has already alluded to from the FISH data, looking at patients with 17p deletions, groups of patients who do particularly poorly. In addition to that, we now know that as well as those patients who have deletions of 17p, those patients who have p53 mutations do equally poorly. So we can identify groups of patients who, at least until recently, we were able to say might be candidates for transplantation. And of course, in the rest of the afternoon, you'll hear why this is all going to turn on its head, because we've got lots of novel agents that may make it much more difficult to determine whether patients should have transplants or not. Dawn has already alluded to this phenomenon, that is, that these p53 deletions and mutations are selected out by the fact that we have treatment. She's already alluded to the fact that this is when people first require therapy. Having some 17p deleted cells might be below the limit of detection of the techniques we've had by things like FISH. We get the therapy, it kills off these cells, but these cells then expand so that what relapses are patients who are going to have a much worse prognosis, and these are the groups of patients that previously we were taking to transplant. So allogeneic transplant remains the only curative treatment for CLL. Hope we won't be able to say that for very much longer. I hope we'll have lots of other things we can add to it. It is a very effective salvage therapy for fludarabine refractory and p53 mutated CLL cells. There's a median survival about 5 years after transplant, although late relapses can occur. And there's very strong evidence of something we call the graft versus leukemia effect. That is, that allogeneic cells from your brother or sister or from a matched unrelated donor are, unlike a patient's own T cells, are able to see and recognize and kill the CLL cells, and that's what we're looking for. There's a but, however. So as we already all know, CLL is a disease of the elderly, and the older you are, the less well you do with transplant. And like you, I've got a different concept of what consists of elderly now as I go on. That comorbidity and not age, we know, is the rate-limiting factor for the use, and the reduced intensity conditioning transplants certainly opened up a window for us to perform allogeneic transplants in a way that we were never able to do when I was first doing transplants, God, 25 years ago, isn't that awful, for using these very aggressive conditioning regimens that people weren't able to withstand. But if you put that graphically here and you think that only 40% of patients are aged less than 75, and that most of those have comorbidities, the number of patients for whom allogeneic transplant could ever be a feasible option remains important, but it still remains small. This is not a good treatment option for the vast majority of patients with CLL, either because the prognosis is too good, or because they have other causes to believe that a transplant will be too risky a procedure. So the factors you have to consider is what's the disease risk, what's the chances that you're not going to respond to other forms of therapy, is there a donor available who's matched enough for us to consider this is going to be done? Are you suitable based upon your age, your frailty, and more importantly, your comorbidity? And what's your preference as to how you want to have your disease treated? And all of these now have to be reconsidered in the context of the new drugs now available. Now, those guidelines were written back a number of years ago, and they were rewritten last year, and I think you already heard Dr. Kaufman talk about the idea is you put 5 experts in the room together to come up with a consensus, you get 5 different opinions instead of a consensus. And right now, I'll tell you, there's very little consensus about where transplant fits in an era of lots of the new drugs you're going to be hearing about later this afternoon. It's really turned the whole thing on its head. But this is the whole concept of how this works. This is a patient that I transplanted when I was still at Dana-Farber, guy who had what's called an ablative allogeneic transplant way back here. And what we're following here are levels of using a very sensitive technique to look for minimal residual disease within his blood. And you'll see that at the time when we do the transplant, his disease goes all the way down, and it doesn't completely disappear, and we're following it. Oops, nowadays we would probably intervene here, but back then we didn't know exactly what would happen with it, and we saw it. You've now finished your PhD. Did you get those last 20 minutes? Let's see if we can get back to it. Everyone's still with me?
Yes.
Anyone had enough? Everyone liked my animation?
Yes.
Good, sorry. I don't know what I'll leave you on. Here we go. Right, so this patient then relapsed, and the only treatment we gave him were donor T cells from his brother. No other chemotherapy, no other treatment. What's happened is his disease goes down, and 10 years later he's still in remission. That tells us that these T cells must have been able to have an effect against the CLL cells. And this is the very early data we had about moving from doing what we called an ablative transplant to what we now call a reduced intensity transplant. Because we reckoned if the T cells could do this, and the chemotherapy and the T cells could do that, why do we need the big doses of chemotherapy? And the answer is we don't. It's the T cells that do the work. And that's the whole concept of what we call reduced intensity transplants. You're letting the donor T cells do the work. Also, again, using samples, we called up Laura, we say we need a bunch of samples of patients with CLL that have got P53 mutations and deletions. Laura provides us the samples. We're able to obtain these donor T cells after an allogeneic transplant, and we're able to sort out a small population of cells that are able to see a specific protein on the surface of the cells. We're able to expand that, and we're able to demonstrate that these T cells can very effectively kill these 17p deleted cells that don't respond to chemotherapy. So they don't respond to chemotherapy, but they can be killed by immune cells. And that's the power of the immune system that we're trying to harness in terms of treating the CLL. So here's data from the kind of the larger groups, the German study group, MD Anderson, Seattle, and the Boston studies, which are probably the largest studies we have in terms of looking at transplantation for CLL, showing us that even in these very poor patients, we can have good survival. But as you're probably also aware, it comes at a huge cost in terms of there's a mortality and there's a morbidity in terms of the risk of graft versus host disease. So we want the graft versus leukemia effect to tackle the CLL, but we don't want the graft versus host disease. And how do we get one without the other? Well, here are your CLL T cells kind of trotting along, and of course what we're looking for, getting onto CAR T cells, we call that the Porsche of the immune system here. And what we're talking about here is taking one of these cells which doesn't work very well, We transfect it with something called the lentivirus, which contains the ability to transmit into the cell, the ability for it to make proteins, which will then be expressed on the surface of the cell. And by putting that antibody attached to the T cell, those CAR T cells are then able to recognize and attack and kill the CLL cell in a way the CLL cells couldn't do. Here's how the process works. People are leukapheresis, the T cells are obtained, they are transfected with that lentiviral virus I told you about. We expand and modify the T cells, we grow those cells up, and we administer those cells back. And what you're looking for here is we're trying to use patients' own T cells that can recognize the tumor, because what we're looking for is to exploit this graft versus leukemia effect without any risk of graft versus host disease. Like all CAR models, there's the 911, the Carrera, and the whatever else model. There's first and the second and the third generation CARs, which are already appearing in clinical trials. There's different targets that these are targeting. CD19 has been the conventional target. Tom's ROR1 antibody, ROR1 protein, is now also being targeted by CARs, and there's other types of CARs that you're going to see. And here's the types of response that you can see. This is data from UPenn on patients who've received these CAR T cells. You're able to see these autologous T cells are able to generate very effective responses in terms of getting rid of these lymph nodes here under the arm to almost completely disappear. And what we see is that the higher levels of these CAR T cells we can find still persisting in patients, the higher the probability of cure, whereas that in those patients in which the cells just disappear, the responses don't last so long. These are not without their own risks. The immune system should be harnessed at your own peril. Immune systems are very safe when it doesn't really do anything, but it can be quite dangerous when the immune system becomes activated. There's a thing called cytokine storm. These T cells expand hugely inside a patient when they're first given, release a whole bunch of chemicals called cytokines that can cause a great deal of difficulty. It's still unclear which model of CAR we should be using and what's the right way to do it. There's the potential for what's called immune escape, that is that these the cancer cells can no longer express the protein on the surface that's the target of the cells, and ways in which we could look at giving multiple CARs together might be the way to do it. And of course, these are also very complex studies for multi-center translation, making it difficult for Bill and I have been trying for a long time to be looking at ways in which we can get CRC-wide studies open on these sorts of approaches, and we will get there. Now, we know that it's the CLL cell that induces the defects. We know what the defects are. We've been doing a lot of work trying to understand precisely what goes on. And in one of our first studies, once we knew what these genes were, which were abnormal in the CD4 and the CD8 T cells, what we were able to do was take CLL cells, obtained from Laura, culture them with completely normal cells, and we're able to demonstrate these CLL cells induce those changes in a test tube. So we know that it's the CLL cells that do it. And we also knew that they required cell-cell contact, so they couldn't be in a test tube with a kind of net barrier in between them. So there was something on the surface of the CLL cell that was expressed that was switching these cells off. And what we were able to do was able to demonstrate that one of the proteins that did this was a protein called PDL1, which, so it's PD1 and PDL1 is something I want to show you a bit more about. What happens on the surface of the T cell as it interacts with either a tumor cell or antigen-presenting cell is there's a whole variety of molecules on the surface of the cell which try to direct the cell whether it should attack or whether it should subside. And there's a whole number of what are called activating receptors, and there's a whole bunch of inhibitory receptors. And whether a cell responds against an antigen or whether a cell responds against a tumor is about the balance of the positive markers versus the balance of the negative markers. And what happens in the CLL cell is that it's got lots and lots and lots of the negative markers and very little of the positive markers. So those negative markers are able to induce the T cells to switch off, which is why you see these very abnormal clapped out T cells. So just shown here, we've got PD-L1 on the surface of the CLL cell, inducing changes, including upregulating PD-1 on the T cells, leading to this exhaustion markers that we saw. It changes the subsets within the T cells. It changes the surface molecules, both within the CLL cell and the T cell. And then there's a complex kind of crosstalk that's happening between these two cells, which are inhibiting each other more and more. It changes the effector function of the cells, it changes their gene formation, and it changes the ability of the cells to see it. And what's basically happening is the CLL has a big stop sign on the surface of the cell, which stops your own body's immune system from attacking it. Now, we haven't been able to get studies yet of PD-1, PD-L1 and CLL, although Bill and I have been trying very hard and knocking on the doors of companies. We are able to treat our CLL mouse model with it to see that this is an important thing we can do. And here's a little study where we took these mice, which were developed within the CLL consortium by Carlo Croce's lab, and we're able to treat one group with a PD-L1 antibody, another group without, and look at the effects that we saw in cells. And what you'll see is, here are the spleens of the mice who didn't get any antibody, and here are the normal spleens of the mice that did. So what you'll see is this PD-L1 antibody is very effectively able to prevent the CLL from being formed, and it's very able to affect the fact that you don't see changes within the T cells. So the future directions and where we're thinking about is trying to refine the role, if any now, of an allogeneic stem cell transplant in an era of novel targeted therapies. Usually when I'm in the clinic now, trying to persuade a patient, well, you can take ibrutinib or idelalisib, or you can have an allogeneic transplant with a 30% mortality. I'm usually seeing the back of something as they walk out the door before I'm going further. We want to be able to look at targeting CLL-specific antigens. So Tom's lab's been doing a lot of work in identifying what these antigens are, so that we've got very specific targets we can go after. We're all working hard to look to improve the CAR T-cell approaches. We have clinical trials that are coming. We've got trials open now of PD-L1 in lymphoma, and Bill tells me he's finally persuaded a company to give him the PD-L1 antibody to CLL to go have PD-L1 trials starting within the consortium very soon in this setting. All of these have the same theme. We are looking to improve the immune response. We're looking to decrease the toxicity of what would be the high morbidity and mortality of an allogeneic transplant, to go for what we're all looking for, which is a cure for CLL. So sorry about the mix up with the slides, but thank you very much for your attention.